Today we will learn about genetic recombination. In bacteria, it involves transfer of genetic information from donor to recipient and result either in substitution of donor's alleles for recipient alleles or addition of donor genetic elements to recipient genome. Transformation, transduction, and conjugation are parasexual process that uses different mechanisms to introduce DNA into recipient bacteria. Here, because DNA can't persist in the recipient bacterium unless its parts of replicon, non-cellular entities are called as replicon, like viroids, in plasmids also. Recombination between donor and recipient genome is often required to produce stable hybrid progeny. Recombination is most likely to occur when the donor and recipient bacteria are from the same or closely related species. Nucleoid plasmid and conjunction occur in transduction, bacteriophage, and bacterial DNA and transformation in naked DNA. His transformation, first mechanism of gene exchange to be discovered in 1928, Fred Griffith found that one form of pathogenic pneumonia, now called Streptococcus pneumonia, could be mysteriously transformed into another form. Griffith's experiment based on fact Streptococcus pneumonia makes two different types of colonies, one pathogenic and another non-pathogenic. Pathogenic excrete polysaccharide capsule that make colonies appear smooth on a garblet. Because one capsule allows them to survive in a vertebrate host, these bacteria can infect and kill mice. However, rough colony can't make capsule and non-pathogenic in mice arise from smooth colony former. In his experiment, Griffith mixed dead Streptococcus pneumonia cells that made smooth colonies with live non-pathogenic cells that made rough colonies and injected the mixture into mice. Mice given injection of only the rough colony forming bacteria survived, but mice that received a mixture of dead smooth colony former and live rough colony former died. Griffith isolated live smooth colony forming bacteria from blood of dead mice. Here is bacterial type effect in mouse and bacteria recovered. Live type R effect non-pathogenic bacteria recovered none. Bacterial type live type S effect pathogenic bacteria recovered live type S. Heat killed non-pathogenic none. Mixture of live R and heat killed S pathogenic live type S. Ayer concluded that transforming principle changed the live non-pathogenic rough colony forming bacteria into pathogenic small colony. Transforming principle was the polysaccharide itself. After 16 years, Griffith did with mice, Oswald Avery and his collaborator used in vitro synthesis to purify transforming principle and showed it is DNA. The work of Avery and colleague helped that DNA, not protein or other vector, is hereditary material. Hence, DNA is a hereditary material. Process of transformation. Transformation of genetic transfer between bacteria in which donor DNA is cell-free DNA and recipient bacterium immediate environment. DNA naturally released into environment when cells die and subsequently lyse. Once free DNA is brought into cell, it is integrated into recipient genome by homologous recombination. Recipient cells that undergo this process and acquire a new phenotype to be transformed. Allow the uptake of DNA from same or related species. It was discovered in Streptococcus pneumonia and occurred in bacteria including Haemophilus, Neisseria, Bacillus, and Staphylococcus.